Yeah, Meiosis Grill. That seems like a really good place to eat. Yeah, man, let's go inside. Yeah, can we, get a table for two, please?
Yes, right this way. Do these seats look good?
Oh, yeah. These would be great, thank you.
Yeah, no problem. What will you be ordering today, sir?
Yeah, this, dish, Meiosis one, that sounds pretty good. I'll have that.
Alright, right away. And for you?
Yes. I would like the, Meiosis 2 dish. That would be great, thank you.
Alright, sounds good. Chef!
What do you want?
We got an order for Meiosis 1 and 2.
Meiosis 1 and 2. Roll the TV show! Hello and welcome to Making Meiosis with me, your host, Chef Meiosis. Today we will be making Meiosis 1 and Meiosis 2. So, here are the ingredients for Meiosis 1. The first ingredient is interphase. Interphase is where the chromosome replication takes place. Each replicated chromosome has two identical chromatids joined at the center. So, you take your first ingredient, or all the other ingredients, and place it in the pan you will be cooking with. Just like that. Your second ingredient is prophase 1. Prophase 1 is where each chromosome pairs up with the homologous chromosome. The pairing up forms a tetrad, containing 4 chromatids, as you can see. Then crossing over takes place. The chromatids of the homologous chromosomes cross over one another and exchange alleles. And your third ingredient is metaphase 1. This is where paired homologous chromosomes line up across the center of the cell. As you can see, these are lined up across the center. Now for your fourth ingredient, anaphase 1. This is where spindle fibers pull each homologous chromosome pairs toward opposite ends of the cell. I know this is taking a while, but please bear with me until we get to the cooking part. Now for your fifth and final ingredient, telophase 1, which will then convert into cytokinesis, which will make your two daughter cells, forming Meiosis 1. Telophase 1 is where nuclear membrane forms around the two clusters of chromosomes. And here, watch this. We're going to just place our last ingredient in the pan. And what you want to do is you want to cook all these on a medium-high temperature. And voila! Now that we have our fabulous Meiosis 1 dish made, we have to make our Meiosis 2 dish. Here are the ingredients for that. Our first ingredient in Meiosis 2 is prophase 2. Now in prophase 2, chromosomes consisting of two chromatids become visible, and they also do not form tetrads because homologous pairs were already separated. So now for our last three ingredients, metaphase 2, anaphase 2, and telophase 2 are pretty much all the same as Meiosis 1. Metaphase 2, chromosomes line up across the center of the cell. Anaphase 2, paired chromosomes separate. And in telophase 2, nuclear membrane reforms and cells separate. Once you have all your ingredients in the pan, you need to turn your stove to a high temperature, knowing that there is double the amount of cells. As you can see, the result of Meiosis 2 is a little bit different. You are left with 4 haploid daughter cells. Each cell has half the number of chromosomes as the original cell, and all 4 haploid cells are genetically different.
Here is your Meiosis 1 dish. Here is your Meiosis 2 dish. So, how's the food, guys?
Oh, it's actually been really good. This Meiosis 1 dish is so amazing. Yeah, your food has been really good. Like, this is the best experience I've had at a restaurant.
Wow, thanks for eating that Meiosis grill.